Welcome, everyone. Thank you for joining today's webinar. The topic today is Computer System Readiness for Platelet Bacterial Risk Control Strategies. For today's webinar goals, we will discuss FDA guidance background and timeline, new testing methodologies, review of bacterial risk control strategies, product codes and labels, and planning for implementation. Some of you may already be receiving pathogen-reduced or LVDs platelets and may be aware of some of the stumbling blocks and software issues encountered along the way. We will focus our discussion on apheresis platelets collected in plasma or PAS. However, some of the bacterial testing strategies also apply to whole blood-derived platelets. Since 2012, the FDA has held 3 Blood Products Advisory Committee meetings to address bacterial risk in platelet transfusions. Their goal was to come up with alternate strategies that would mitigate risk of bacterial contamination and ultimately prevent septic transfusion reactions. The result is this final guidance, Bacterial Risk Control Strategies for blood collection establishments and transfusion services to enhance the safety and availability of platelets for transfusion. I'm sure, like me, most of you have read and perhaps reread the guidance several times. So why is this guidance so significant? This is probably the biggest wholesale change we have seen in blood processing in a long while. Some statistics that influence the guidance are listed here. Room temperature platelets are associated with higher sepsis risk and related fatalities, and bacterial contamination of platelet products is the leading risk of infections from transfusions. It is believed that 1 in 10,000 to 1 in 100,000 transfusion-transmitted infections per year are caused by contaminated platelets. Evidence suggests that 1 in every 6,000 products are suspected to have an infectious agent. What is most significant is that the highest risk of transfusion-transmitted infection occurs in platelets that are transfused on days 4 to 5 of their shelf life, with 95 to 100% of septic transfusion reactions occurring on these days, and 100% of fatalities within the same range. This risk has continued even with the implementation of a single culture at 24 hours after collection. In December 2018, the draft guidance for bacterial risk control strategies was first released. By September 2019, the final guidance was released with an implementation date of March 31st of this year. Because of the COVID-19 pandemic, implementation of the guidance was delayed from March 31st, 2021 to October 1st, 2021. Fortunately, this gave both blood centers and transfusion services a little more time to plan and prepare for implementation. In preparation for this guidance, the Blood Products Advisory Committee needed to determine when was the best time to culture, how was the culture to be performed, and if a second culture was needed. They also wanted to investigate alternate strategies to mitigate bacterial contamination risk. Strategies needed to be implemented that would not limit the shelf life of platelets, but would ensure that platelets stored beyond 3 or 4 days were safe to transfuse. The following strategies were introduced. Primary testing using large volume delayed sampling technique, or LVDs. Secondary testing by culture or bacterial detection by an FDA-approved rapid test, and the use of pathogen reduction technology, or PRT. There are at least 7 pathways to implement bacterial risk strategies. Some are single-step strategies and some are 2-step strategies. The single-step strategies include 2 methods for large volume delayed sampling, one with a 5-day expiration and one with a 7-day expiration. Single-step strategies also include pathogen reduction technology. This would be your sorrelin-treated platelets. Two-step strategies are a way to safely extend the life of the platelet by either using a secondary culture or using rapid testing. Primary cultures will be performed by the blood center, but hospitals may perform secondary cultures or rapid bacterial detection as part of their two-step strategy. Most hospitals prefer the single-step strategies as there is really nothing more to do once the product gets to the transfusion service. However, shelf life, cost, and product availability can be limiting factors. In some rural areas that may be a long distance from blood centers, more flexibility may be desired, and those hospitals may opt for a strategy that will accommodate greater shelf life or shelf life extension. This graphic displays the life cycle, if you will, of platelets with single-step strategies. The time of collection on day 0 will determine how soon the product can be sampled for culture or undergo pathogen reduction. You can see that each product may be ready to release from the blood center at different times, even if the collection is on the same day. The arrows represent the approximate length of time platelets can be stored once they reach the blood bank. The number of days available for transfusion is variable, as receipt at the hospital will be impacted by the time of collection, the time of processing, or the operations of your blood center. During the planning stages, it is very important to discuss inventory concerns with your blood supplier, especially if you are limited to certain types of products. Note that some products may require a hold time if shipped prior to the end of culture incubation time. In those situations, inventory controls must be in place at the transfusion service. Let's take a closer look at some of these one-step strategies and how they differ from current methods. For LVDs 36, the platelet is cultured no sooner than 36 hours after collection. A 16 ml sample is evenly divided between both aerobic and anaerobic cultures. Each container from a split must be cultured separately. The minimum incubation time is 12 hours, making the total hold time at least 48 hours after collection. Although the initial expiration is 5 days, the product's shelf life can be extended using the appropriate FDA-approved secondary testing. Here is an example of a product label from a single-step LVDs 36 product. Note the text bacterial monitoring greater than or equal to 36 hours. The expiration will be 5 days from collection at 23:59. Below are some examples of product codes that fall into this group. You may notice that these ISBT 128 product codes have the new EA prefix. Your donor or transfusion service computer systems may require a software patch or upgrade in order to use or receive these EA codes. If you are modifying or dividing and printing labels, your label printing software may also need to be upgraded. Be aware that ICBA is rapidly creating new product codes. About 600 EA codes have been defined since September 2020. LVDs 48 is similar to LVDs 36, but the platelet is sampled for culture no sooner than 48 hours after collection, with the same 12-hour incubation time. The 60-hour hold time at the blood center seems like a long time, but these products will have an overall shelf life of 7 days from collection. This is a popular option for its extended shelf life, but may not be desirable for certain populations or physicians that require fresher platelets. It's also important to note that the guidance requires that platelets must be collected in a container that is FDA approved for 7-day storage. Here is an example of a product label from a single-step LVDs 48 product. Note the text, bacterial monitoring 7 days. The expiration will be 7 days from a collection at 23:59. You may also notice that some of these product codes are the new EA codes as well. Keep in mind that these codes are just examples, and the product codes that your blood center will be sending may be different from the ones listed here. Before you start updating your computer system, make sure you have a current list of products and E-codes you'll be receiving from your blood supplier. Pathogen reduction, or pathogen inactivation, or PI, is the process of treating the blood components soon after collection in order to inactivate any remaining infectious agents. Although the technology is termed pathogen inactivation, the components themselves are referred to as being pathogen reduced. Pathogen reduction uses a chemical agent that is activated by UV light to bind nucleic acid so that DNA cannot replicate, and thus the cell cannot replicate. We know that red cells and platelets do not have nucleic acids, but lymphocytes do. Therefore, their proliferation is prevented by PI treatment. This process is effective against many infectious agents, including viruses, bacteria, parasites, and protozoa. Pathogen reduction technology has been shown to inactivate meaningful titers of key viruses and other infectious agents, including HIV, West Nile virus, Zika, Chagas, and parasites, including those that cause malaria and babesiosis. Pathogen reduction is performed within the first 24 hours of platelet collection. It can be used on both platelets stored in plasma or platelets stored in platelet additive solution. Sorolin is first added, then the product is submitted to UVA light. The sorolin is then removed from the product using an absorption device prior to distribution. This technology has been widely used since its FDA approval, and more blood centers are offering pathogen-reduced platelets in addition to other bacterial risk control strategies. So what are the benefits of PRT? Because infectious agents are pretty much destroyed, it greatly reduces the risk of transfusion-transmitted infections, including cytomegalovirus. Therefore, CMV testing is not necessary for treated components. It also eliminates the need for irradiation to prevent graft versus host disease. This has caused somewhat of a logistical puzzle in some computer systems, where the software is checking for CMV negative and irradiated attributes to meet patient transfusion requirements. Some of our BCS clients have created workarounds for this issue, or simply adjusted SOPs to include internal controls to override the computer warnings that display. I would not recommend adding a CMV negative attribute to pathogen-reduced platelets unless it was truly tested and displayed on the label. The same goes for the irradiated attribute, as irradiation is not recommended for these products. But there are some considerations if you are thinking about using pathogen-reduced products. They are contraindicated in patients that are sensitive to sorolins and in neonatal patients being treated with phototherapy. Although they may arrive at your blood bank a day sooner after collection, pathogen-reduced platelets cannot be extended to 7 days using secondary testing. The cost to manufacture PI products is a bit higher and may cost more to keep in inventory. However, some say the cost may balance out if you end up wasting less product. Here is an example of a product label from a PRT platelet. Note the text sorolin treated displays. The expiration will be 5 days from collection at 23:59. In your computer system, you may want to consider adding a pathogen-reduced unit attribute to these products as part of their unit record, especially if you are overriding CMV negative or irradiated transfusion requirements. Although not listed here, many of the sorolin-treated product codes will have an EA prefix, especially if you are configuring open codes to be used for aliquots and syringes. Let's review two-step strategies. A two-step strategy is a method by which a blood center or transfusion service can extend the shelf life of a platelet, but still minimize the bacterial risk. In the first row, the primary culture is performed at 24 hours. This is still allowed by the FDA guidance. But to comply with the bacterial risk control strategies, the blood product will require secondary testing to be transfused after day 3. Use of a second step must be performed to achieve the full 5-day shelf life. The second row, LVDs 36, will expire at the end of day 5 unless secondary strategy is performed. For step 2, either a secondary culture or rapid testing can be performed for either of the step 1 products. If you are using daily rapid testing, it must be performed within 24 hours of transfusion up to 7 days. It is important to note that a transfusion service, even if currently exempt, but is now implementing extension to day 6 or 7, are no longer exempt and must register with the FDA. First, let's look at the product label where the primary culture was performed at greater than or equal to 24 hours. These platelets will be initially labeled with a 5-day expiration. However, the label must clearly indicate that retest is needed after day 3. If secondary culture is performed, you can relabel or simply cross out the retest statement. Alternately, you can wait and perform a secondary culture at day 4 using a 16 ml sample and 12-hour incubation to extend the out date to 7 days. When secondary rapid testing is performed, relabeling is required to indicate testing at day 6 or day 7. Expiration time must also be included on the label. Many will ask if rapid testing must be performed if the unit is not transfused within 24 hours. The simple answer is no. However, it may be difficult to bring an expired product back to life in your computer system once it has expired. I would try this in your test environment before you write that into your SOPs. To assist in planning, the AABB conducted a survey from March 23rd through April 5th of this year. The respondents included 272 hospitals, some with on-site donor centers, as well as 22 dedicated blood centers. This group represented about 20% of total annual platelet transfusions and about 76% of total U.S. collections. 32.8% of respondents indicated that they were already in compliance with the FDA guidance, while 67.2% planned to be in compliance by the October 1st deadline. 66 of transfusion services preferred to carry only one type of platelet. 29 hospitals had already implemented rapid testing to extend out dates to 7 days. Many of the respondents expressed supply and demand concerns, while some were now considering using low-yield platelets in order to maintain inventory levels. Interestingly, the top 3 platelet products preferred by transfusion services were pathogen-reduced platelets in PAS, 7-day LVDs platelets, and pathogen-reduced platelets in plasma. Yet the donor centers listed LVDs 7-day platelets as their most offered product, followed by LVDs 36 and pathogen-reduced in plasma. It's probably safe to assume that the types of products offered by blood centers will continue to evolve even after the October 1st deadline, as blood centers continue to roll out a variety of products. Transfusion services will need to be prepared for product options that are not currently available from their blood supplier. Earlier this month, AABB published a bulletin to its members which laid out the impact the FDA guidance will have on donor centers and transfusion services. These 5 key points were stated. 1. Mitigation strategies must be in place by October 1st, 2021. 2. All mitigation strategies improve the safety of platelet transfusions. 3. All mitigation strategies are approved by the FDA and deemed therapeutically equivalent. 4. Hospitals should be prepared to accept a range of products to maximize platelet availability. 5. Hospitals should be prepared to perform secondary testing to maximize platelet availability. They went on further to say that these strategies, PRT protocols, new product codes and outdates, and labeling changes will require both vendor and in-house IT staff to support, modify, and validate regulated software. Let's review some of the planning components you may need to help you get to the October 1st deadline. Transfusion services must be prepared to receive a variety of new platelet products. Communicate with your blood center to make an implementation plan. Be prepared to alter the plan if needed. Perform new product build in your blood bank computer system. This may include product modifications, additional attributes, test build, and billing. All new build must be verified and validated prior to implementation. Computer systems and printer software may require a code upgrade or a patch to allow entry of the new ISBT 128 EA codes. Contact your software vendor for details and an implementation plan. Use of sorrelin-treated, pathogen-reduced platelets will impact how patient transfusion requirements are met in your computer system, especially for those patients who require irradiated or CMV-negative products. Donor centers will require changes to platelet manufacturing processes, depending on what bacterial strategies they are using. Inventory levels of desired products may be impacted. Distribution plans will likely need adjustment. Many hospitals will have dual inventory of pathogen-reduced and LVDs platelets. Inventory controls and storage should be addressed. Will separate shelves or platelet rotators be used for different products? How will you keep your inventory separate? Will rapid bacterial testing be implemented? Determine modification labels to be used. Consider staff training and updates to SOPs. Determine if submissions to the FDA will be needed. Who will be assigned to file and follow up on these submissions? Will education of medical staff and nursing on pathogen-reduced products and extended outdates be needed? Updates to your Circular of Information brochures may be required. How will implementation of bacterial risk mitigation strategies affect your operating budget? Will it cost more to maintain your current inventory levels? Additional cultures, rapid testing, labels, printers may all add cost. Implementation requires expertise, information, time, and expense. That concludes my presentation for today. I would like to acknowledge AABB, ICBA, ARC, and various blood centers around the country for the wonderful job they have done in keeping us informed on these bacterial risk mitigation strategies. Below are the references used to prepare this presentation.